Good afternoon, and thanks, Michael, for inviting me again to this symposium. So I'm going to talk about cars, and Michael put the little phrase in front of my title about immuno-oncology cars, because he didn't want people who were not interested in Ferraris to leave. So I'm going to talk about a Ferrari car, but not necessarily an automobile. But I thought I could use the car-automobile type analogy to describe some of the phenomena that are occurring now in the field, as Dick mentioned, a very exciting area in immuno-oncology. So I'm going to talk about CAR T cells of the Ferrari type. And if you can imagine having a Ferrari automobile, it's got a huge engine, but if it had no brakes and the accelerator was stuck to the floor, it has only one speed, and it's full speed ahead. And that's where the Ferrari CAR Ts are right now. Well, that's very interesting, and as long as you can point it at the right target. The problem is that with the Ferrari type of CAR Ts, they have a steering wheel, but it's locked. And so it's straight ahead at whatever that target is. And I will point out to you the problems that this creates in clinical medicine, clinical oncology, and what we think at AvidBiotics, a subsidiary of which now is called Zyphos, which is our immuno-oncology subsidiary. We're going to describe convertible CAR T cells, not a Ferrari convertible, but a convertible CAR T cell. And the question is, what for? Why do you need a convertible CAR T cell? Well, the New York Times has published a number of articles over the last 5 years on CAR Ts. And as they say, the CAR Ts are essentially one of the means of harnessing the immune system to fight cancer. So, what's a T cell? Christoph gave you some idea, he mentioned extensively, actually, T cells. So T cells are one type of lymphocyte, which is one of the types of white blood cells that circulate. And the T cell, the T stands for thymus derived. The T cells have a lot of receptors for various antigens on their surface, but every CAR T cell, or every T cell, not a CAR T cell yet, but every T cell has a receptor for only a single antigen. But because you've got many, many millions of different T cells circulating, there are many, many receptor-bearing T cells for different antigens. The T cell is shown here in the context of a red cell and a platelet. So that's what a T cell is. What's a CAR T cell? So a CAR T cell is a T cell that has been modified genetically. These are T cells taken right out of frequently the patient that you want to use these subsequently in. And if the T cells in a person's body doesn't have the right receptor, there's not a T cell with an appropriate receptor for a tumor, then that tumor is going to grow into a malignant tumor because there's no means of containing it. But if you can identify a receptor that recognizes the antigen on the tumor, and that is personalize it, and you can take the gene for that receptor and couple it to some other machinery, intracellular machinery, you can introduce that gene for what is called a chimeric antigen receptor, which is where the CAR comes from. Introduce that gene into this T cell, and you end up with a CAR T cell. And it has on its surface a receptor that's encoded by this are, or this receptor, that now is targeted or steered directly straight ahead at the tumor antigen that you would like to use to kill that tumor cell, and the T cell will attack it if it can find it and bind to it. Okay, so that's the CAR T. And this is a cartoon showing a little of the mechanics of this. If you have a a cancer cell that has a tumor antigen on its surface, and then you take a CAR T cell, this is the T cell, and this is the chimeric antigen receptor. It's got some internal machinery that will activate this cell, and then the receptor, or the chimeric antigen receptor here, is in all of those that are, at least in the approval process at the FDA now, they're single chain SVs from antibodies. And so this is the antigen recognizing component of an antibody that has been fused to this internal machinery. And so what do you use these CAR Ts for? Well, you take them out of the patient, you make them CAR Ts, you take the T cells out, you make them CAR Ts, and then you reinfuse them into the patient. And the problem is that there's no steering wheel, you're straight ahead, and you can only target a single antigen, whatever happens to be, whatever receptor you have picked to target this particular antigen. But as I'll show you in a minute, that creates problems because frequently the cancer cell will become resistant by simply shedding that tumor antigen. And then your receptor is useless. And you have to start all over again. You need a new CAR for every new target. The other issue is that in this situation, it's always on. So we've got a situation with no steering wheel and no brakes. So you put this into a patient, and what happens is with no dose control, because you can't control the dose of these because they're going to replicate once you put them back into the patient, then you end up with no dose control, and the consequences are you end up with enormous, what's called a cytokine storm, release of cytokines, interleukin, TNF-alpha, IL-6, et cetera. And these are responsible for the symptoms you get when you get the flu. But with the cytokine storm, it's the worst flu you've ever had in your life, and it may kill you if you've gotten such a release of cytokines that you're in a storm. And then if the receptor here happens to recognize, not on the tumor, but on some normal tissue, the same antigen, for instance, suppose it's against HER2, which is on a third of the breast cancers or a quarter of the breast cancers, et cetera, but it's also in the heart. And it may be more densely populated on the cancer, but in the heart it's there. And so if you've got something that's sort of not controlled, it may cause a lot of heart damage. But you want to be able to turn it off, but you can't turn it off, and you can't turn it off because it's full speed ahead. The other problem with this, as I've already mentioned, you've got a single target that is on this, that this receptor recognizes. And thus, when a tumor escapes by shedding the antigen which it's seeking, then you end up, again, with toxicity that's quite significant, and this is essentially the straight-ahead problem. So this is the problem with the conventional CAR Ts, but let me add, they are incredible if they're targeted right and you need a full speed ahead CAR. And they're curing patients at least long-term survival 50 to 60% in patients that were stage 4 melanoma, for example, or leukemias. All right, so where do we go from here? Well, we have at AvidBiotics and now Zyphos worked in the pathway which is called the NKG2D MIC-A pathway, and I won't go through the name, where this came from, but this ends up being part of another lymphocyte called an NK cell, which is naturally circulating, and it has a receptor on its surface called an NKG2D receptor. This is all just a natural receptor. And this receptor binds a ligand which is not normally expressed on cells, not on healthy cells, but on cells that are stressed. This ligand and several other ligands that are recognized by this receptor will be expressed. And when this NK cell, which is roaming around your blood and tissues, sees this ligand on a surface of a cell, it binds it, it becomes activated, and it'll kill that cell, that stressed cell, because it's a threat to the host. So what types of stresses? A viral infection, a malignancy, radiation, hypoxia, all of those stresses can induce these ligands. And the T cell, as a major surveillance system, goes around and zaps those cells. And it's when they escape, when, for instance, a tumor cell escapes by shedding this ligand, that one can get a cancer because the surveillance system can't see it. Okay, so we've been working in this pathway for a number of years by genetically engineering or protein engineering this ligand so it bound really tightly to this receptor. And we found that, at least in mice, we couldn't make it work the way we wanted it to because the NK cell can't get hot enough in terms of its activation and killing in a solid tumor. So a colleague of mine, Kyle Landgraf, who came out of protein engineering group at Genentech about 5 years ago, was working in this pathway, and I don't know, he had a nightmare or something happened, and he had this paradoxical idea. Why don't we break this receptor so it can't see any of these ligands? And then he said, and I can think of how we could use it. Now, at that time, the receptor and its ligand their structures were known in 3 dimensions at high resolution by a group in Roland Strong's lab at the Hutch. And what was known specifically that Kyle focused on was there is a tyrosine residue at Y152. There are 2 tyrosine residues that are key for this interaction. And without belaboring this, what he did was to change this Y to an A, to an alanine. And we already knew from Roland's work that that was going to completely kill the ability of this receptor to bind to the ligand, any of its ligands. And so what he did was to inactivate this receptor, and so it's changed its ligand binding motif here, and it can't bind any of the wild type or natural ligands. Well, what good is this? Well, we knew enough, and he knew enough about this domain, which is the binding component here, to, with some phage display, to go in and make mutant alpha-1, alpha-2 domains that can no longer bind the wild type receptor, but can bind the otherwise inert receptor. And so he created an ability to pair in a privileged way a ligand with this inert receptor. And if he put an antibody onto it, we created what we call a microbody. So this now creates a very different situation. And that is, if we make a CAR T cell out of this inert receptor and put their internal machinery on it to activate this CAR T cell, and the resting state is completely off. It doesn't see anything that it knows. Even if the stress ligand were on this cell, it doesn't see it. And it can't see the tumor antigen because it's not designed for that. It's designed to be inert. But then, when you put in a microbody, it turns it on. And so this gave us a way to activate this convertible CAR T cell. And not only that, but it also gave us a way, excuse me, to do multi-antigen targeting, either sequentially, excuse me, or multiplexing. because there are plenty of these receptors on the convertible CAR T cell. So this certainly changed the situation, and rather than show you a lot of work that went on in vitro and some early in vivo work to prove that this phenomenon actually works in vitro, targeting a specific cell that has the right ligand on, that has the right antigen on it. And if the antigen isn't there, you can't activate the cell. And when you activate the CAR T cell, you get cytokine release and you can kill the cell. It will attack the cell. Rather than do that, let me just show you some animal data, which is jumping, a couple of years of work, essentially, to give you some bottom lines and some images. So this model that we have used is taking NSG mice, and these are severely immunodeficient animals, so they're not going to reject human cells when we put them into the animal. So we're doing what are called xenografts. And the cell that we are using, the tumor cell, is a human Raji cell, which is a EBV transformed human B cell malignancy. These Raji cells have been modified to contain luciferase, which some of you I'm sure are familiar with, is the enzyme that fireflies use for converting luciferin into actually a flash of light. And so by putting luciferase into these cells, we can actually monitor, as I'll show you, how many cells are around and where they are in the animal by just pictures. So we gave these animals 500,000 Raji cells IV on day 0, and at the same time, we gave them either 4 micrograms, 20 micrograms, or 100 micrograms of a microbody that has as its antibody anti-CD20, and CD20 is a surface antigen that's present on this Raji cell. So we've matched the receptor with the microbody that only sees the inert receptor, and the other end of which sees this cell. And then we introduced the CAR T cells the day later, and we gave them 5 million. And they were both CD4 cells, which you heard about earlier, and CD8 cells in a 1-to-1 ratio. And they came out of a healthy human. And then the animals were continued to receive once every 4 days, thanks Michael, once every 4 days another dose of each of the 4, 20, or 100 micrograms. And I went the wrong way here. And then we imaged them once a week. And the results are shown here. So if you just give these Raji inoculated NSG mice just the vehicle as a control, they're essentially all dead at 2 weeks. If you give them just the highest dose of this microbody, which is using rituximab fused to the light chain, and it's this orthogonal, as we call it, ligand, U2S3, you can see that they too are dead at 2 weeks. The T cells, if you give these animals the inert NKG2D CAR, at 2 weeks they all look very sick. There's a little activity here which I can explain to you, or Patricia can explain to you. And the untransduced T cells, that is exactly the same T cells coming out of the same human, do the same thing. So these are truly inert T cells, even though they have a CAR structure in them. And the microbody itself is inert, but you put them together, and these are the animals, the cohort that received 4 micrograms, and then 20, and then 100 micrograms of the microbody, followed for 3 weeks. And what you see is that the 4 micrograms helped. They survived this long. The 20 micrograms looked better, but the 100 micrograms look worse. And so we have an inverted U dose response curve. Not unusual And the immunologists call this this phenomenon a prozone. And what is happening there is that you're saturating your CAR T cell with the MicroBody, and so all of its receptors are bound up. And your target cell has all of its antigens, the CD20, bound up. And there's no beachhead for the CAR T, the bispecific MicroBody, to grab both ends and pull them together. And that's why at high dose you begin to lose activity, a well-known phenomenon. All right, so this was evidence to us that we now can control activity by dose. We have to get the dose right, and we can control the targeting, because unless the CAR T is in the presence of the MicroBody and a target, you don't see any activity. So we did another study, and this is just further exploring the dose optimization. And this dosing was same dose of Raji cells, and at the beginning, they got either MicroBody, the same MicroBody, either every 2 days, Q2D, or every other day, or every 4 days. And so we're increasing the frequencies from every 4 days to every 2 days to see whether that makes any difference. And then we gave 2 different doses of CAR Ts. So we're increasing the frequency of the MicroBody delivery, and we're doubling the number of the CAR Ts. So what's happening here, in effect, is that we're increasing the size of the engine here, and we're stepping on the accelerator from here to here. And what happens is interesting. Here's the same vehicle type of pattern that you see. Here are the inert CAR T cells at 10 million. And you see it's really inactive. By the time these animals have died, these are nearly dead, but then they get a little recovery. And the untransduced T cells act the same as these. So these are really, really inert. They're inert as pure T cells from the same donor. But look at the dose response now. And that is with 5 million CAR Ts, and every 4-day dosing at 20 micrograms, you see more or less what we saw previously. And if you increase the dosing, it looks a little bit better. You give it every 2 days rather than every 4 days. If you double the horsepower and increase the CAR Ts to 10 million with every 4-day dosing, you get still better activity. But if you do both, you essentially are knocking out the tumor. And so this is just further evidence that you have control with dose, in this case, dose of the inert CAR T cell and dose of the MicroBody based on the frequency. So this is why we think this approach solves some of these problems of the Ferrari CAR Ts, and yet also retain the properties that the combination of a CAR T targeting and dose control can provide, at least to NSG mice. And just very quickly, let me just tell you where we are. We're essentially headed to the clinic for a first-in-human proof of concept. We have follow-on indications that we intend to pursue other than CD20. CD20, you might know, is very close, not just in number, very close in terms of its expression pattern to CD19. And almost all the activity that you've read about, at least in the New York Times, as well as much of it in the literature, is targeting CD19 on B-cell leukemias and lymphomas. So we want to move out of that into other epithelial cancers rather than B-cell malignancies. And then what I think is really important is that this seems, at least in mice, to be quite safe with this control and ability to retarget. But there are lots of patients who could benefit from a CAR T if it were just safer. And so that, we think, opens up the possibility of going into patients who have a lower risk tolerance than a patient with cancer on their deathbed, which is the way these Ferrari CAR Ts have been used pretty much to date. And a key one, which you heard about just before lunch, is antiviral, and specifically HIV, not to kill the virus, but specifically to attack the CD4 cells, CD4 T cells, that are latently infected with HIV. Because if such patients who are controlled and suppressed by the hearts are taken off the drug, they suddenly will start producing virus from these CD4 cells. And there is an approach now where people are trying to use antibodies and what's called shock and kill, and that is you shock the CD4 cells so that they start to release a little bit of virus, and then you kill those cells as soon as they show their head. Because as you may know, HIV is essentially silent in the CD4 cells. They don't put anything on the surface that anything can recognize. But if you can kick them or shock them, and they start to stick out some envelope proteins, then you ought to be able to kill them with a CAR T. But the CAR T has to be safe because these patients are being well managed, as you heard, on their antiviral drugs. And then that opens up autoimmunity and inflammation, et cetera. So we think there are a lot of opportunities for CAR T cells, particularly if they're safe with control. And so without belaboring this, we essentially are trying to create a precision CAR with control and multi-targeting that has accelerators and brakes. Navigator we need, diagnostics, to identify the target for which we want to build a MicroBody. We don't have to rebuild the CAR T cell or the CAR. We can use the same one for every target and every disease once we know the target. And we have a steering wheel for personalizing. And so what we're trying to avoid is this. And we think we're on the way. Thank you.